In this video, we explore how biologists organize the immense diversity of life by looking first at why we classify and how the traditional Linnaean system groups species. We then connect this to modern molecular evidence, cladistics, and finally, we analyze cladograms to interpret patterns of relatedness. These key concepts are some of the most important topics covered in the IB biology syllabus. There are millions of discovered species on Earth. Many of these species give important clues about ecosystem health, climate change, and evolution, while others have important compounds for medicine. Just like we need a common system of measurement for sharing data, we must also classify these organisms to aid in research and communication about different discoveries. When a new species is discovered, classifying it gives us important information about its metabolism, morphologies, reproductive strategies, and many other characteristics it shares with closely related species. Classification systems should also be able to tell us about their evolutionary relationships, including species that are closely and distantly related to them and recent common ancestors. For instance, if a newly discovered aquatic animal has a blowhole instead of gills, we can use classification to determine that they are likely more related to whales than sharks. This in turn allows us to draw other conclusions. They likely nurse their young, need to surface periodically, and may form social groups. The traditional classification system developed by Linnaeus uses a hierarchical organization of different levels of taxa based on physical features or morphologies. Taxa, or a taxon, is a group of organisms that can be grouped together. Taxa can be very broad or very specific. The broadest category in which all life can be organized is the domain, which has 3 taxa in it: eukaryota, bacteria, and archaea. The basis for this organization is on similar characteristics, like the presence or absence of membrane-bound organelles or differences in their cellular walls. Domains can be divided into further taxa. The eukaryotes is divided into 4 kingdoms: Animalia, Plantae, Fungi, and Protists. Then Animalia can be further divided and so forth, with each level becoming more and more specific. For instance, the Chordata phylum only includes animals that have a notochord in embryonic development. In the phylum Chordata, the class Mammalia includes animals that have a notochord and also have mammary glands that produce milk, as well as hair or fur. The order Cetacea includes all the previous characteristics, but only include animals that have forelimbs that are modified into flippers. The family Balanoptera have streamlined bodies, pointed snouts, and long grooves on their throats that allow them to capture large amounts of food. The genus Megoptera includes the additional characteristics of long, large pectoral fins, bumps on the head and body, and unique tail fin patterns. The classical Linnaean system has 8 levels of organization, with genus and species being the most specific. In this example, M. novianglium is known as the humpback whale species, who can only mate and have successful offspring with other humpback whales. A species' scientific name includes its genus and species, with the genus often abbreviated. As more and more species have been discovered, so has our technology and ability to objectively analyze relationships between species. Using RNA, DNA, and amino acid evidence has led to discoveries about evolutionary relationships and has caused us to reconsider some of the ways we've organized and categorized species, leading to reclassifying some species. As a result, leveled hierarchical systems have needed more levels and sublevels added to account for new discoveries. For instance, in previous classifications, we had 5 kingdoms, and the Monera kingdom included all prokaryotes. However, once new technologies became available and genetic analysis of microorganisms were possible, it was discovered that some prokaryotes share a more recent common ancestor with eukaryotes than they do bacteria. A new level of domain was added above the kingdom level to account for this new discovery, and we now have the domains of eukaryota, archaea, and bacteria. If we were to look at the domain taxa and organize it into a cladogram, we would see that the archaea are more related to eukaryota than they are bacteria. This was a major shift in classification, but smaller shifts have also occurred, leading to further modification of the traditional classification, such as adding subphylas and superphylas. Organizing species into a leveled hierarchical classification system is useful but limited. Because it uses morphological information, it does not always reflect evolutionary relationships. The hierarchical structure does not consider the gradations of relatedness that we see when species slowly evolve over time, or when genes are transferred between different species of bacteria, or when hybridization of plants or animals occurs. Cladistics is a different system that aims to visually organize species based on evolutionary relationships instead of hierarchies. Cladistics allows us to place species into an evolutionary context to determine their relatedness to other species, as well as place them in a broader evolutionary context. When new species are discovered, they can easily be added to an existing cladogram using shared or derived characteristics that indicate a last common ancestor. For instance, the Mun River alligator is an extinct species that was discovered in 2023. It has a short, broad snout that placed it as closely related to the Chinese and American alligator. The Mun River alligator also has a small opening in the roof of the mouth, a raised ridge on top of the skull, and an elevated ridge behind the nostrils, which the Chinese alligator has, but the American alligator does not. Using these shared and derived characteristics allows us to conclude that the Mun River alligator is more closely related to the Chinese alligator than the American alligator, and should be placed in the cladogram to show these relationships. Moreover, for living species and very well-preserved species, cladistics can use more objective data like differences in genetic sequences, such as DNA or RNA, as well as differences in amino acid sequences. This gives us more objective data and measurable ways to classify organisms that is based in evolutionary relationships rather than the more subjective practice of using morphologies. Cladograms can be thought of as a series of nesting clades. Clades are a group of organisms that have arisen from a common ancestor. Because evidence suggests that all life arose from a single ancestor, known as the last universal common ancestor, or LUCA, all life on Earth can be considered as one clade. Practically, scientists and researchers are usually interested in clades of a much smaller scope that consider either individual species or different taxa. Here, we're looking at several taxa of different classes. Let's consider a cladogram consisting of the classes birds, mammals, lizards, lungfish, flounder, perch, spiders, and lobsters. The tips of the branching point where species or taxa are named are considered terminal branches. So lobsters are on a different terminal branch than the spiders. Branching points are called nodes, which indicate speciation of a last common ancestor into two or more groups of species. A clade is any group of organisms that share a common ancestor. So a cladogram is actually many nested clades. This whole diagram could be considered one clade. However, lizards, birds, and mammals would also be considered a clade as they share a more recent common ancestor. A larger clade could also include lungfish because they share a common ancestor with lizards, birds, and mammals. However, flounder, lungfish, lizards, birds, and mammals are not a clade as they don't share a common ancestor. Perch would have to be included to consider it a clade. Clades can be switched around, so positioning the bird and lizard in opposite positions is also correct, as well as flipping around the entire diagram. You can think of it like a solar system mobile, where the overall structure stays the same, but the clades can rotate around their nodes and still display the information accurately, like the planets can rotate around the sun but still display their correct order. Each branching point in a cladogram is called a node and represents a hypothetical last common ancestor. Because fossil evidence is incomplete, these last common ancestors are hypothesized. We do not have fossil evidence or other evidence to know their exact morphologies. The branching at the node indicate a speciation event where the last common ancestor was reproductively isolated and split into two or more species. The base of the cladogram that includes the last common ancestor for all the species represented is called the root. Although common ancestors are hypothetical, we can infer the characteristics they must have possessed from the traits shared by all descendants in a clade. For example, in the clade that includes lizards, birds, and mammals, each group has an amniotic egg. Therefore, this characteristic must have evolved before their last common ancestors speciated into these lineages. The amniotic egg is a primitive or plesiomorphic trait of this clade that includes the amniotes. Lizards and birds can see UV light, whereas mammals cannot. Based on this distribution, where should the ability to see UV light be placed in the cladogram? If you placed it here, before the common ancestor of lizards and birds, but not after the common ancestor of lizards, birds, and mammals, you're correct. In this clade, the ability to see UV light is a derived or apomorphic trait because it evolved after the last common ancestor speciated. As a result, only some members of the clade possess this characteristic. Although we have considered a clade using primitive and derived characteristics, differences in genetic sequences can also be used to construct cladograms. When comparing genetic information, DNA, RNA, or amino acids can be used to compare the similarities between species. Because DNA codes for RNA, which codes for amino acids, and the differences between genetic information build as populations speciate, this is a reliable and quantifiable way to measure the relatedness between species. Using genetic sequences can also give an estimated time of speciation as mutations occur at a relatively even rate. This is what is referred to as a molecular clock. And although it's not precise, it can give an estimated time frame of speciation. If more mutations have accumulated between two different species, they are less related versus species that have had less differences accumulate. Cladograms will often display a number indicating the differences in either a DNA, RNA, or amino acid sequence, and this information is valuable in building cladograms. If we look at an example here, we have several different species with a genetic sequence that has been aligned. We would count the differences we see between each species and place that information into a table. So for instance here, species A and B have 7 nucleotide differences in the DNA sequence in the gene that we're comparing. We would do this for each species, counting the difference in nucleotides between each to fill out the rest of the table. We would then start with the species that show the fewest differences, and therefore are the most related. Species C and D have the fewest base differences with only one change, and so we would place them close together, showing their close evolutionary relationship. We'd then look for the next fewest differences, which is between species E and C and D, with 4 differences in their sequence. We then find the next most related species, which is species B, with 6 differences from species E. Finally, our outgroup with the most differences in their genetic sequences from the other species is the least related. Because mutations occur at a relatively predictable rate, these speciation events could be set against a timescale that estimates when common ancestors diverged and speciated. Often, the genetic evidence between species verifies the evolutionary relationships that were derived from using morphological features. However, occasionally genetic data leads to reclassification of groups or species. The Figwort family were traditionally grouped using morphological traits such as flower estivation and nectary structure. But these features turned out to be misleading because unrelated plants had evolved similar characteristics, or analogous structures, through convergent evolution. Beginning in the mid-1990s, DNA analyses, especially comparisons of the nuclear ribosomal ITS region, revealed that the traditional Figwort family was not monophyletic. Its members did not share a single recent common ancestor. Instead, the group was paraphyletic, with species spreading across separate branches of the evolutionary tree. As a result, botanists reclassified many figworts into new families that reflect true evolutionary relationships. A well-known example is the foxglove, once considered a figwort, but now placed in the plantain family. This reclassification illustrates how genetic evidence can overturn morphology-based assumptions and produce classifications that better represent evolutionary history. In this video, we reviewed classification and cladistics. Below is a summary of the key points that we covered in this video.